Where am I supposed to be? Can you see me? Buenos días a todos y a todas. Good morning, everyone. Welcome to the EAP Monteverdi Institute Simposyum. This is the culmination of about 10 weeks of work, really more than that, because many of you started thinking about your independent projects a year ago, or some of you in high school. So, we're off to a great start. We have lots of fascinating, great talks today. Just to explain part of the dynamic, the moderator will introduce you, so you don't have to say the title of your talk and your name again. You can if you wish. The moderator, who will be sitting here or here, will flash you with a card that says you have 5 minutes left of your 9 minutes, and then at the appropriate time, the moderator will flash this card which says you have 2 minutes of your 9 minutes left. And then, should have drawn a happy face on this. This is, you're at your 9-minute limit, okay? And in the ideal world, you would leave us 3 minutes for questions. One thing about questions is that the instructors will likely cool their jets and let students and members, other members, invited members of the audience to ask questions, okay? So we'll cool our jets and let members of the non-instructor audience ask first. Good morning, Silvio. How are you?
I'm great.
I'm great. All right. Silvio is a graduate of the University of California at Los Angeles with a PhD in classics.
Not a graduate. I did most of the program.
Okay, good. Excellent. Bueno, we're ready to start, and yeah.
Morning, everybody. So we're going to start with our third session about living things in the mangroves. So our first presenter is Simposyum.
Hi, good morning, everyone. My name is Millie. I'm presenting about mycorrhizal presence and abundance in mangrove forest in UCEAP, Costa Rica. So first, just a brief introduction on what mangroves are. They're a really critical ecosystem that, despite only covering 0.1% of Earth's surface, offer a huge amount of ecosystem services that benefit all living organisms. So first off, they sequester a lot of carbon. They also provide storm surge buffering, pollution filtration, and can protect large amounts of marine biodiversity. So my research mainly focuses on 4 species that are present in large amounts in the UCEAP area. And those are Avicennia baculare, Avicennia germanans, Rhizophora mangle, and Loducularia racemosa. So another brief background on what mycorrhizae are, since that's what my research focuses on. Mycorrhizae are mutualistic symbiotic fungi that connect plants and their substrates by entering roots and establishing hyphal networks. I have a brief little diagram here on the 2 types of mycorrhizae that affect mangroves. So first we have on the left ectomycorrhizae, and then on the right, we have arbuscular mycorrhizae. And the main difference is whether or not they actually penetrate the cell wall of the root cells. So the benefits of having established mycorrhizae in plant roots are numerous. They allow increased access to nutrients. They increase tolerance to heavy metals and salinity. They make plants less susceptible to root pathogens, and they also just generally increase plant resistance to biotic and abiotic stressors. So my central questions are centered around one regenerative mangrove plot. In UCEAP, I was curious because mycorrhizae are very sensitive to disturbed soil and high salinity soil. I was curious if they were present or not. So my first question was, does mycorrhizal fungi colonization occur within the root systems of mangroves after generating soil flats? And then my second question was, is there a discernible variation in the percentage of mycorrhizal colonization based on the different salinity levels and substrate types observed among 4 distinct mangrove species present in Kimikio? So on to my sampling locations. I chose 2 locations, Kimikio and the regenerating salt flats. Kimikio is shown on the left and has salinity percentages that range from 34 to 35. The regenerating salt flats are shown on the right, and the sampling locations have soil salinity levels of 25 to 54%, so a much broader range. I conducted my sampling between November 26th and November 29th, 2023, and I took root samples from juvenile trees from 15 different trees at the 2 different locations. And so I took my samples from sources that were less than 3 meters from the water source, which was either a river or a canal in the case of the salt flats. I use a refractometer to measure groundwater salinity that I used as a proxy for soil salinity. And yeah, on to my next methods. So in order to actually examine the roots under the microscope, you have to clear and stain them. So I cleared my roots with 10% KOH by boiling them in that for 15 minutes. Roots that were still pigmented, I let soak for another 8 minutes and then rinsed thoroughly in distilled water. After that, I boiled the roots in 5% ink and vinegar solution for 2 minutes and then thoroughly rinsed them. After that, I mounted them on slides. I used 3 1x5mm sections from each root. I labeled and then relabeled the slides so that I didn't know what slides I was looking at in order to decrease potential bias in my study. Next, I scored each root sample that I was looking at based on the following scale. So 0 was no mycorrhizae present, 1 was 1 to 5% colonization present, 2 was 6 to 25, 3 was 26 to 50, 4 was 51 to 75, and 5 was 76 to 100%. So the mycorrhizal elements that I observed were mostly of 3 types. First and most abundant was the dark septate endophytes, which I'll refer to as DSE. The second was DSE mycosclerotia, and the third was AMF hyphae. And these are in order of the frequency in which I observed them. So I had 30 root samples. Out of all 30, they all had evidence of mycorrhizal colonization, which in and of itself is very important and indicates a strong association between mangrove roots and mycorrhizae. So I calculated the average colonization here and graphed it against the species, and I found that there was a statistically significant difference between the average colonization percentages, and I ran a Krustal-Wallace test and got a p-value of 0.024, and found that Avicennia bicolor had the highest mean colonization level of 2.074, plus or minus a standard deviation of 0.77. So having a heightened relationship between Avicennia and bicolor and mycorrhizal fungi is important because it shows that that species has a higher affinity for the mycorrhizae, and this is potentially important because it contributes to its success in the Santillana Gulf. This species in particular is very abundant and very resilient in these high salinity areas. Next, I examined substrate. So I divided my data into 3 categories based on the soil types that I observed in the samples, which was mixed, mud, and sand. And then I calculated the average colonization level for those categories. I ran a Krustal-Wallace test and found significance with a p-value of 0.021. And I also analyzed the preference by species. So I found that Avicennia bicolor preferred mixed and sand substrates. And yeah. So overall mixed, like all of the species tended to have a preference for mixed soil, which could be significant because the porosity of mixed soil is greater than in just mud, which allows for increased root growth. But in comparison to sand, it has more access to organic matter, so there's more nutrients available for both organisms. And it is possible that specializing in specific soil and salinity types offers some sort of resource partitioning between these species, which is important since they don't have anything in a shared area. Finally, I looked at salinity, and the only species that showed a relationship between salinity and average colonization of mycorrhizae was Avicennia bicolor, and it had a strong preference here for salinity ranging between 32 and 45%, which is fairly high. So this suggests a potential reliance of Avicennia bicolor on mycorrhizal fungi to thrive within these high salinity zones. And one potential reason that it doesn't extend much past this zone is that high salinity is often fatal to mycorrhizae, and so it would be less observed. In conclusion, mycorrhizae are found throughout the salinity gradient, which is super important because it means that mangroves are able to grow in these areas, and in the salt flats, it means that the inoculation methods that they're using have been successful and could be replicated at other regeneration sites. Furthermore, the fact that DSE are more abundant than AMF may mean that they're more adept at dealing with high salinity and could be used in further research. And more research does need to be done to identify which species of mycorrhizal fungi specifically are present in these soils. Yeah.
We have time for questions. Questions for me?
So, are there a lot of different mycorrhizal species in the world or in the area, or do they tend to have it in certain places?
Yeah, there are tons of different mycorrhizal species. So within both of the categories that I mentioned, there's multiple thousands into the tens of thousands.
Do you think that those survive at different salinity levels?
Definitely.
Nice. Okay.
Do you know at what point that mycorrhizae become present?
At what point they become present?
Yeah.
In terms of salinity?
No, in terms of plant growth.
Plant growth? They can colonize fairly quickly. I don't know the exact time frame that they need though.
More questions for me? Fran?
There's been lots and lots of attention paid to zonation in mangroves. What's your sense with respect to the relative importance of propagule, dispersal, and access to mycorrhizae?
Well, access to mycorrhizae is super important in the zonation because it influences how much access that these propagules will have to certain nutrients and how resilient they will be in the face of climatic stressors. So increased mycorrhizae in general is a huge benefit to different species.
Time for one more short question.
What was the most fun part of your project?
Definitely, I forgot to go to my acknowledgement slide, but I got to bike to some of my sampling locations. So you can see here, blue and behind us is Avery, and then our guide Medaya, who helped us bike all the way to the nucleus.
Thanks
So,
for being
thank you for the story.
So our next speaker is Tia.
medical progression, yet we continue to allow neglected tropical diseases, specifically the vector-borne ailments of Chagas disease and dengue fever, persist in the most vulnerable communities in our world. In fact, the continued progression of these diseases only serves as a testament to the failures of our global medical system. In the absence of adequate research, these diseases spiral unchecked. It's already known that frequent dengue outbreaks strain localized healthcare communities. So for example, they exacerbate wait times, they exhaust resources, and they financially devastate low-income households. It's estimated that there is 400 million people on this planet who get infected with dengue per year. And yet, despite this, our global understanding of the disease remains to be grossly neglected. In terms of Chagas disease, it is estimated that there's 8 million people on this planet who currently suffer from the disease, and that 50,000 of those people will die per year. Chagas disease is transmitted through contact with the infected feces of the Triatima dimidiata, otherwise known as the kissing bug. And if this parasite, Trypanosoma cruzi, successfully infiltrates the host, it will cause silent havoc, slowly turning into a long-term disease called chronic Chagas cardiomyopathy. And in 40% of those people, they will suddenly die of a tragic heart attack. Now, I decided to focus my research in Guayaquil because unlike a more developed region like San Jose, for example, Guayaquil does not have the privilege of a well-equipped and advanced healthcare system. In fact, Guayaquil suffers from a lack of patient education, limited healthcare access, and very limited disease surveillance. So the truth is, our understanding of dengue transmission and Chagas disease in Guayaquil is very limited just because we don't know much about it. There's very little research done in this specific area. And that leads me to my research question, which is, what is the population proportion of Aedes mosquitoes, which are the primary genus of mosquitoes that transmit dengue fever? And what is the prevalence of Chagas disease in Guayaquil? So as I mentioned, dengue fever is transmitted by Aedes mosquitoes, primarily Aedes aegypti and Aedes albopictus. And what's interesting about dengue is that it's considered to be an urban disease. So where there are more people, there are going to be greater incidences of dengue outbreaks. Now there's also 4 strains of dengue that is known currently, and your body can only gain immunity to the strains that it's been exposed to. What's also important to consider about mosquito-borne viruses is that there's the cycle of the disease in the mosquito and also in the human. So a mosquito gets infected with dengue when it bites a person that is actively febrile, and then it sucks up those viral particles and it goes into the gut intestine of the mosquito. And then 8 to 12 days later, it will transport up to the salivary glands, and that is how someone else can get infected with dengue. So when that mosquito inevitably goes to bite someone else, it will transmit those viral particles from the salivary glands into your body. And you'll start to show symptoms about 4 to 7 days after you were bit. So what did I do to actually study dengue in Guayaquil? Well, I caught mosquitoes. And I did that by, whenever I saw a mosquito, I would spray it with a bottle that had soap and water solution. And then when that mosquito fell to the ground, I would pick it up with tweezers and then place it into a labeled plastic bag. And I did that in 3 primary locations: the Mangroves, Restaurant Artesipe, and Casa Verde. And I also did that throughout different times throughout the morning, afternoon, and evening. So in total, I caught 117 mosquitoes, and 26 of them turned out to be belonging to the Aedes genus, and I was able to identify 4 of them down to the species level, which was Aedes albopictus, which is a verified dengue transmitter. So another thing that I found from this research is that the highest prevalence of Aedes mosquitoes was found near Casa Verde and then the mangroves. And interestingly, 0 Aedes mosquitoes was found at Restaurant Artesipe. And this is depicted in the geographic heat map on the right, and then the pie chart on the left. And as you can tell, the greatest proportion of Aedes mosquitoes was found at Casa Verde. And I speculate this because around this area, there's the most people living there and the most potential breedable habitats. So what's interesting about Aedes mosquitoes is that if you left out a tire or a pot and it filled with water, Aedes mosquitoes could breed there. And there's an abundance of those habitats surrounding Casa Verde. Now this chart depicts at what different time intervals I found these Aedes mosquitoes. And the biggest takeaway from this is that there were no Aedes mosquitoes found at night. Most of them were actually found between the times of 9 to 10 a.m. and 5:30 to 6:30 p.m. And I did run a chi-square test on this, and it showed that there was a statistically significant correlation between the time of day and when Aedes mosquitoes were present. So now I'm going to transition to how I study Chagas disease. So with the assistance of 9 different people, we surveyed 15 different locations, and it accumulated to a total of 45 hours of human effort. And ultimately, this yielded 1 Triaxoma dividiata. An example of the types of places we looked at were private farms, abandoned farms, private residences that had livestock living there, and even the local vicinity of the supermarket. Surprisingly, the 1 Triaxoma dividiata we found was by the local supermarket, and I suspect that this was there because in that area there was a lots of stray cats and dogs, so I speculate that this particular bug was feeding on those stray animals. So in order to see if this Chagas bug actually had Chagas disease, we had to examine its species. So what I did, or what Frank and I did, is we laid it on its back on a glass slide, and while Frank held it with metal forceps, I used a metal probe to massage the abdomen and move in a downward motion. And eventually, when the fecal sample came out, we placed it under a compound microscope, and using oil immersion, we observed the feces using 1000 power. So this particular Chagas bug was in fact positive for Chagas disease. And we know that due to the presence of the parasite Trypanosoma cruzi. And we know that this is the parasite because of the distinct long and narrow flagella that is used to facilitate its movement. So taking this all into account, I devised 3 main methods of preventing the spread of dengue and Chagas disease in Coahuila. So the biggest and arguably the most important aspect of these prevention strategies is education. So I would suggest implementing programs that educate the population of Coahuila on how they as an individual can protect themselves and what they can do to recognize Aedes mosquitoes and destroy those habitats. And the second aspect of this would be tailored awareness programs towards those high-risk households. And a previous study discovered that a high-risk household for dengue is a household that has difficulty recognizing symptoms, a household with lower education levels, and a household that has higher incidences of dengue within their home. And finally, the third aspect is continued disease surveillance, because with more data, we can create more tailored approaches. And finally, I'm just going to briefly touch upon telemedicine and its potential to reduce health disparities in rural areas. And this is based off of a study I read that was about implementing telemedicine in a rural community in Mexico. And I believe the benefits could be translated to Coahuila if we were able to overcome the obstacles. So some of this includes increased accessibility, so people would be able to access a healthcare provider at any point in time. And this would also reduce travel time and expenses because an area like Coahuila has limited transport options to their nearest hospital, which is in fact almost 50 kilometers away. And then finally, there's increased healthcare efficiency because there are less people in these waiting rooms, and there's less chances of you getting infected by something else just by being around so many sick people. I truly believe that telemedicine could be the long-term solution to a whole multitude of diseases in Coahuila. And finally, I'd like to thank my primary advisor, Frank Joyce, and my secondary advisor, Federico Chinchilla, as well as any of my peers and the residents of Coahuila that helped me in our research efforts. Thank you.
Questions for Jordan?
Yes.
So the second highest abundance of dengue mosquitoes was in the mangroves, right?
Yes.
So why do you think that, like, is there something for them to feed on in and around there, more so than like restaurant Arrecife, which has a lot of people?
Well, not every single Aedes mosquito feeds on people, so only the females can do that. But I suspect that there are a high prevalence of Aedes mosquitoes in the mangroves because I observed a lot of abandoned boats or buckets that would fill up with water, and those I believe would be adequate breeding sites for Aedes mosquitoes. And there's also people that live in the mangroves, like on the outskirts of it.
Right.
Bryn?
Do you have any ideas why the dengue-carrying mosquitoes, you didn't see any of them at night?
Well, there is this preconceived notion that Aedes mosquitoes are only active at night. It could just be that they are diurnal, but there's also studies that show in the presence of light, even in the middle of the night, that those mosquitoes will be attracted to the light. Daniel?
Is there a way to test the mosquitoes themselves for dengue?
Yes, there is, but it's a really complicated process that involves tracking the disease particles within the mosquito itself, and it would take a lot of time and effort in the actual lab. Yeah. It uses inflorescence as well. Yeah. Haley?
Did you look at the percentage of mosquitoes that you caught that were male versus female?
Yes, 55% of them were female, and all 4 of the Aedes albopictus were female. Yeah. Jake?
Are there any biocontrol methods that are used to control Aedes populations?
Biocontrol methods. Actually, so there's a lot of studies on this right now. So I mentioned that it takes 8 to 12 days for the gut particles of the virus to travel up to the salivary glands of the mosquito. And if you were able to somehow stop that process, you could theoretically end dengue transmission. So people are studying that right now on ways to control the cycle of the virus in the mosquito as a means of protecting people. Rachel.
What you just mentioned, would that like deal with kind of halting the virus so it doesn't get to the mosquitoes like proboscis, if that makes sense?
Yeah, so somehow they would prevent the disease from transporting up to the salivary glands, because if it just stayed in the gut of the mosquito, when it went on to bite someone else, it would not inject those particles into the person.
Cool.
Thank you, Jordan.
So this study was conducted in Guayaquil, Costa Rica, where we observed this mysterious glow in the soil, and it was actually really cool to observe. It was almost like you were looking at like a night sky full of stars but at the ground. And then we later figured out that the cause of the glow was bioluminescent firefly larvae. So bioluminescence is the production and emission of light from a living organism as a result of a biochemical reaction. And its function varies greatly through nature. Some examples can be for mating purposes, for interspecific communication, and as a lure for prey, but it's not known for all animals. Firefly larvae in particular are super understudied. In fact, a literature review found that 94% of species in the family Vampiridae, which is the family of all firefly larvae, have never been studied as larvae, and over half the genera, which is pretty crazy. And in particular, the function of their bioluminescence is widely disputed, which is why I chose to study what I did. And the observed mangrove firefly larvae that we saw in Guayaquil are potentially the species Micronaspis florida due to morphological similarities. These are some pictures, both taken in the field and through dissecting microscopes. Excuse me. So as you can see, they have this really cool ability of retracting their heads completely into their bodies, as you can see right there, and right there. And
that's actually what their head looks like. It's super weird, they're really long. And this is just a close-up of their scales on the ventral side of their bodies. Almost looks like dragon scales. And now that we're all a little more familiar with the study organism, I want to introduce my central question, which is, does the bioluminescence of mangrove factfly larvae function as an anti-predatory defense? So my predictions are that A, the larvae will be more likely to glow, and also will glow for longer when exposed to stimuli of higher intensities that mimic the attack of a predator, and also that predators will avoid feeding from the larvae. So for my anti-predator defense trials, what I did is I subjected the larvae to 3 stimuli of varying intensities. So the first was blowing air through a straw, which was meant to simulate a breeze or just a gentle disturbance in their environment. The second was the stroke of the paintbrush, which was meant to simulate a soft touch that could be mistaken for the touch of a predator. And the last was meant to emulate an actual interaction with a predator. So what I did is I lifted the larvae 5 centimeters above the substrate with soft forceps. So 20 trials were conducted, and the presence or absence of the glow was recorded along with the liquid flow. Excuse me. This is a screenshot of one of the stimulus trials that was filmed. And as you can see, the larvae is glowing in response to the stimulus, which is a stroke of a paintbrush. That's a paintbrush right there, that's a larvae, and those are the lanterns. For the predation trials, I offered my larvae along with alternate prey to geckos, which are generalist predators, easily found in Guayaquil. 10 trials were conducted, 5 of them used fruit flies, and 5 of them used mosquitoes. This graph shows the number of flows elicited by each of the 3 stimuli, and as you can see, for the stimuli that have greater intensity, there are a higher number of flows elicited. This graph shows the average glow time that each stimulus caused, and as you can see, there is a similar trend where the more intense stimuli elicited a higher average glow time. This is a representation of the prey items consumed by the geckos, and for 4 of the 10 trials, neither the alternate prey item nor the larvae were consumed, but for 6 of the 10 trials, only the alternate prey items were consumed. But as you can see by that gap in the middle of the graph, the larvae were never predated on. And this is actually a super cool picture of one of the geckos I used in my trial. And as you can see, he's just hanging out with the larvae. He doesn't look like he wants to eat the larvae doesn't look scared, which is interesting. So as I mentioned before, there are many potential functions of bioluminescence in nature. But if we kind of go back to the stimulus trials, we observed a pattern of there being more glows and a higher average glow time for more intense stimuli. And the stimuli were meant to emulate the touch or attack of a predator. So that kind of indicates that our larvae are glowing in response to an interaction potentially with a predator, and that if they were to interact with a predator, they will also glow. And that can be inferred to mean that their bioluminescence is a reaction to a predator and can be used to startle the predator. For our predation trials, we observed that our geckos did not predate on the larvae at all. And their reluctance to eat the larvae could indicate toxicity, which in turn could mean that the bioluminescence can also function as a warning to predators of their toxicity, like an aposematic warning. And there are a lot of other potential functions, but there are various reasons as to why I believe that they are less likely than the warning of predators. So for the first option, interspecific communication, it's not likely that they're communicating to other larvae because they weren't observed to live in social groups, and also because they're nocturnal insects and they have very small eyes, so it's not likely that they have high visibility at night. For mating, we can cross that off because they're larvae and they don't mate until their adult stage. For mimicry, there is a chance that they're trying to mimic adults that have innate toxins in their blood, but if the adults have innate toxins, there's a good chance that the larvae have innate toxins as well. And as for the lure to attract prey, it's unlikely because firefly larvae prey on gastropods, which have very little visual acuity. In conclusion, the bioluminescence of mangrove firefly larvae likely functions as an anti-predatory defense. And why I believe these findings are important is because there was a recent petition to list Micronopsis floridana under the Federal Endangered Species Act, and it's actually already categorized as endangered on the International Union for Conservation of Nature Red List of Threatened Species. So I believe it is all the more important to understand these creatures in order to better protect them. Also, researchers believe that there are likely undescribed populations of Micronasca species in coastal Central America, and if this larvae is indeed in the Micronascus genus and potentially Micronascus florigana, it'll be the first recorded observation of them in Costa Rica, which is really exciting, and also important to bridge that knowledge gap about their distribution. Thank you to Emmy, to Frank, to Richard, Leo, Josie, Millie, Haley, Avery, and of course, Lilia, my homestay mom, for everything they did. I wouldn't have been able to do this without you guys.
Okay, we have time for questions. Questions for each human. Maybe, do you know how long the larvae usually spend in the larval stage?
No, actually. So the life cycle of firefly larvae in general is very understudied, but specifically this is a relatively new species that's discovered. So no, I'm not sure. And also we're not even sure if it's seasonal because when they were first observed, there were a lot of larvae, and so we were thinking maybe by the time of the home project there wouldn't be any left, but there were still a lot, so no clue, and all different sizes too.
So you mentioned that the head can fully retract. Do we understand why?
Yeah, not sure, but they seem to do it in response to like stimulus. So for example, I have a little terrarium, or I have one in the lab right now where I'm keeping a lot of the larvae, and when we poured water into the jar to moisten it, the larvae kind of retracted their heads in, but they also seem to do it naturally while they walk, so unclear. Rachel?
Could you explain a little bit about the distribution of these larvae within the mangroves? I know at some point you were wondering, are they intertidal or something like that?
Yeah, so the Micronaspis genus is actually colloquially known as the mangrove firefly and the intertidal firefly, so I do think that they are intertidal. I didn't observe them past that one section. I don't know if you came with me, but it was like, if you walk straight from the mangroves towards the end, there was one section where I found a lot of them, and there were a lot past that section, but they were present in spots of the mangroves that would have been flooded at high tide. So I do believe that they are intertidal. And then outside of Coahuila, they're found in Florida, in like the coastal northern Bahamas, unidentified, unconfirmed species in Jamaica, Belize, and Panama, which is pretty cool. And then they just found a new species in this genus in Brazil. Yeah, but all in mangroves, all coastal.
Okay.
And all intertidal.
We have 1 minute more for questions. Any other question?
Frank?
Are you familiar at all with some of the implications of an animal being declared an endangered species, like say in the United States? Like, what does it actually mean for the management and conservation of that species?
Ooh, well, I know what qualifies in insect or animal as endangered, but I'm actually not sure how that would affect how they're conserved in the future. But I know that it has to be, like their distribution has to be limited to like a certain range. I think it was, I want to say 200 square kilometers, something like that. It might have been 500. But then also the fact that they're threatened by like habitat loss and a bunch of other factors like that. But we probably know.
Just in general, what would you say is the sort of status of the Guayaquil mangrove? Is it okay, or is it under assault, or what would you say with respect to your mangrove firefly larvae?
I would tentatively like to say that it's okay, although we did observe a lot of pollution after the Rio Guayaquil was flooded. I think she's in good hands.
Okay.
Thank you, Regina. And he's going to present his work on the non-critical lab.
All right, hi guys, I'm Lou. I'm going to be talking to you about Noctilia leporinus, also known as the greater bulldog bat. This is a resident of the Quahinaquil mangroves. It fishes in the streams and rivers there. Bats have this incredible ability to produce echolocation calls which are adapted to their spaces that they're in. This is incredibly important for these fishermen bats because fishing in an aquatic environment rather than hunting terrestrially is a much different game to play. For the purpose of my investigation, I've kind of distinguished two different classifications for streams. One is narrow and one is wide. The narrow is less than 15 meters in width and the wide is greater than 25 in width. Here's a sample of an audio call that I've analyzed. In orange, you can see the constant frequency portions. These are the portions with the highest frequency. It's listed on the y-axis. There's no dramatic spike or drop in this call. And this call is very unique to the Noctilio. And then in the black dotted portion, you can see the FM tail. And the FM stands for frequency modulated tail. And this means the length of this tail is what discerns how much information is received by the bat per call that's released. So using those width classifications, I'm led to the question, how does the microenvironment influence the abundance and acoustic variation of Noctilio leporinus echolocation? To get into my methods and materials, I had a lot of fun. I was able to kayak through the mangroves to set these devices called audio moths, as you can see up here in this corner. They're very small devices. They allow us to analyze echolocation calls with really incredible precision. I started with two primary locations. I began in Rio Quahinaquil, and I recorded 2 nights at 2 different sites per night, one narrow and one wide each evening. I configured these moths to record from 5:30 p.m. to 8:30 p.m., which is the time known to have the highest activity of Noctilio leporinus. And so the way that I conducted this is I kayaked out at high tide. I placed the moths at 2 meters above the water at high tide level to keep it consistent across my measurements. And then while I placed them, I recorded the width using a transect sort of tape situation that I fashioned out of a measuring tape, and I just measured the entire width of the chain. And then after the 2 nights at Rio Quahinaquil, I moved on to Rio Murcielago, which was a bit different in setup. It was a lot shallower, so I didn't have to kayak out. I was able to just place it in the evening and collect it that same night. So for the first kind of parameter that I'm analyzing, I'm looking at the activity of calls. And this is represented through the abundance, or just the number of calls that I was able to detect using my audio moth. On the x-axis, we have the narrow and wide channel treatments, and then on the y, we have the mean number of detected calls per night. There's not a huge difference in these trends. Both have pretty significant error bars, suggesting that there's pretty high fluctuations. We have a mean of 29 calls per night occurring in the narrow, and then in the wide treatment, we have a mean of about 23. Since there's no kind of statistical significance in the difference of occurrence of calls in the environments, I'm led to further pursue the acoustic variation parameters, which I will get into next. This is suggesting that Noctilio can produce different kinds of calls, different FM tails, based off of the environment that they're in response to vegetation and clutter. So this segues into the first of my acoustic parameters. My methods in this was to manually go through every single call that was a hit for Noctilio leporinus and find the cleanest calls. I chose...
The electricity is going to go on and off. Maybe we can use this.
Mm-hmm.
Is it plugged in?
Mm-hmm.
Is it
Is
on?
that the one? Mm-hmm. Estaba aquí. That was my plan. I was making a break.
No, not yet.
Ahora, es que la extensión está conectada. O sea, no debería ir.
Entiendo, sí, pero puede ser como que...
Pero pasémoslo para acá. Creo que sería mejor. Pero es que a veces... O sea, a
No, la laptop no. Conecta solo el proyector porque...
El grupo técnico está trabajando arduamente en resolver ese problema. Pronto estaremos continuando.
Sí. Sí.
Todos los cuatro.
Sí, son los 4. Y uso solo para el proyector porque si no, no va a arrancar. Ahora chocamos. Y las compus tienen sus baterías. Y ya debería. Y ya eran solo listos. Pura bien.
No se va a ir.
No, no, voy a estar aquí.
Sorry for the technical issues. We are going to continue.
Okay, so back to my first acoustic parameter, which was the maximum frequency. It was really important to streamline the way that I chose my calls, so I chose to only analyze FM calls. Remember, that's the one with the tail that drops off. The reason to do this is because it includes both the CF and FM components, and that is going to be able to show us the maximum frequency and duration for the entire call. So looking at the maximum frequency, there's a clear gentle trend towards higher overall maximum frequency in the wide treatment versus the narrow treatment. And then this is measured in kilohertz per second. I conducted a 2-sample independent t-test to conclude that there is very high statistical significance between an increase in maximum frequency channel versus a lower maximum frequency in a narrow channel. Intuitively, having fewer obstacles, as you can imagine in a wider channel, would mean that there is more room, more time to be perceiving things that you were hunting. This kind of increases the search process, makes it more rapid, and just enables quicker hunting practices when there is less clutter in the area. The next parameter that I want to talk about, and the final factor that I considered in acoustic variation, is the duration of each call. Again, I exclusively use these FM calls because the tail is the part that drops off and kind of provides the most accurate view of a duration of a call. There's not a huge kind of visual trend here. The one thing that I really noticed is that there's a lot more of a range in the narrow channel versus in the wider channel. It's a lot more precise. And I conducted a third 2-factor independent t-test, and that kind of concluded that there was a moderately significant correlation between the increase of the duration of call that occurs in a wide channel versus a narrow channel. A potential explanation for this is that a longer call can support kind of increased echo detection. So when a call is sent out and then it's sent back, if the call is longer, you're going to receive more information coming back to you. in open spaces, this kind of increases the bat's ability to recognize the spatial kind of composition that they're working with. To conclude, this is the first study done to investigate the acoustic parameters of Nectilia labrinus in mangrove environments. It has provided insight into how the maximum frequency and durations can change, and it kind of provides insight into their resilience and cooperation with the environment. So it indicates that there is a difference detected in their environment, but there's no deterrence in the way that they're foraging. On a broader scale, this is really exciting for me because it can provide a little bit of a clue into how these bats might evolve and change with time, as they can change their communication and change their foraging practices to best fit an environment that is changing with them. And then finally, I'd just like to thank everybody who helped me. There were so many of you. Thank you to all the EAP contributors, collaborators, everyone who was with me in Creaking the Keel. Your support was invaluable. Thank you guys.
Speculation on how you think that the acoustic parameters of this species would change in either environments affected by anthropogenic development or in the future of like climate change, how they might differ in comparison to now?
Yeah, so I think primarily it would change with prey availability in these brackish water channels. Mangroves are kind of estuaries. They're sanctuaries for these breeding fish that are growing up there. The fish that they're preying on are very small in size. So I think with fluctuations in the availability of prey, they're going to have to seek out other areas. That means that they might be more inclined to enter more narrow channels, more areas with kind of greater shelter that can provide space for more fish as those fish species seek in. As for the anthropogenic side that you mentioned, I did take measurements in both kind of a more densely human populated area on Rio Quijiniquil and then also a more isolated area in Rio Murciélago. So I kind of got both sides of that coin. So my findings are kind of an average of the two, but it could be really interesting to compare the two and keep them separate.
Very nice project. Do you know if this bat species shares the channels with the lesser fishing bat? And if they do, can you distinguish them in your recordings?
Yeah, so I can distinguish them in the recordings. I only focused on the greater fishing bat, but the albiventris, the lesser, was present in working with one of my buddies, who will present very soon. We experienced both bats in the same channels, so she will get to that. I'll leave that to her.
So you can distinguish. How do you know which is which in the recordings?
I just use the software to help me distinguish.
Okay.
The software is Kaleidoscope. I'm not sure if I mentioned that.
Okay. Thank you.
More questions for them?
So I'm guessing that you were kind of daunted by the complexity of a mangrove habitat and marvel at the way the bats managed to navigate through it. But I'm wondering what else they might use in addition to sort of instantaneous echolocation cues about how to avoid this or that obstacle. For example, do you think that they might make mental maps and know what's a good foraging area, a clear foraging area versus what's one that is really dangerous? And then the other thing is, why not just always fly out over open water and not mess around in a tangled, dangerous place?
The condition. Okay, so to answer your second question first, the conditions are very different in open water. There's a lot more factors. It's a lot less sheltered. So you're kind of factoring in wind and a lot choppier waves. Those are just factors that you don't need to experience if you're in a more protected mangrove stream environment. Secondly, yes, mental maps are very important. There have been many studies that show that when mist netting traps are put out and kind of other environmental factors are changed, there are recollections of that, and that changes kind of like the passing through that their life happens.
Awesome, thank you.
Thank you. So our next speaker is and she's going to talk about, again, the beautiful Castillo de Perú.
Hello everyone. I'm excited to share my research with you today. For my research project, I decided to research the sexual dimorphism in the wings of the greater fishing bat from Guayaquil, Costa Rica. So to start, I'd like to introduce bats. So bats are mammals, if you didn't know. So that means they have live births. They breathe air. They're mammals. They're also of the order Chiroptera. So that means that literally means winged hands. So they're pretty famous for their membranous wings, and so that's why they're called as such. So what a lot of people don't realize about bats is that bats do a lot of work for our environment, right? So they are pest controls, they pollinate, they disperse seeds for us. They're a lot like birds and bees in doing the same jobs, except they work at night. And then these are a couple of the different species that are here in Costa Rica. So I'd like to introduce wing morphology because it is what I chose to research. Wing morphology is pretty much how the shape of the wing can affect the flight of something. So similarly to how different types of planes have different wing shapes, so do bats and so do birds. So here is an example of two different bats. So you can see two different species of bats. So you can see in the top photo, this bat has very kind of short, rounded, thick wings. And in the lower photo, this bat has super elongated, pointed wings, right? And this is because they have different foraging patterns and because they're different species, right? So the top species is carnivorous. So this species eats insects and small invertebrates. So they have to be really fast to catch an insect. I don't know if you've ever tried to catch an insect. I know Jordan has, but you have to be fast, right? So you have to be super fast, especially if your prey is on the ground. You have to be able to hit the ground, pick up your prey, and then get off the ground in order to not hit you. So you have to be really fast. That's why they have those short, thick wings. And then this lower species, which is the fishing bat, which I researched, they have these really elongated wings to help them like glide, right? So they're a lot slower, and they have much more like gliding patterns versus the quick darting patterns. So within wing morphology, there are different measurements you can use to kind of give it a numerical value. So one of these is wing loading. And so what wing loading is, it's the mass of the object, so whether that is an animal or a plane or a bird or a flying thing. Anyway, so you take the mass, you multiply it by gravity, and then you divide it by wing area. So I am going to explain this a lot more simply, but what is important to understand is that the smaller the value is of the wing loading, the more the vessel can carry, because the wings are larger than, the wings are larger relative to the mass. So let me explain. Okay, we have two bats, okay? The wings are the same size. Okay, so I also took out units and gravity to make it a lot more simple. So the wings are the same size here, right? They're both 200. However, the mass of the two different bats are different. One of them is significantly larger than the other. One is 600, one is 400. So if we solve for wing loading on the further left bat, we take the 600, the mass of the bat, and we divide it by the wing area. By 200, we're going to get 3. If we do the same thing for the other bat, we're going to get 2. It is a smaller wing loading value for the bat on the right. That is because the wings are larger relative to the body, okay? So lower wing loading, larger wings, it can carry more, okay? We're gonna revisit this. So, the species I chose to research is the greater fishing bat. These bats are very, very cool and very tricky. So they eat fish. And a couple of things about them that makes them really unique that they have developed to be able to fish is they have giant feet and they have these giant wings. So you can see here, they have these giant feet that they use to just like scoop up fish from the water. So as we learned from Lou's presentation, they echolocate throughout the water, they sense a fish and they pick it up, right? They also are like bright orange with this like racing stripe. They're really cool. So my proposed question was, what are the differences in wing morphology between the sexes of the greater fishing bats and Coahina keel, specifically investigating wing loading? I expect lower values in females. So I expect that the females are going to have a lower wing loading, meaning that they're going to be able to carry more. Lower wing loading, they're going to be able to carry more. And I will explain why I thought that later. So, methods. I was in Coahina keel for the 2 weeks, which there's Coahina keel relative to where we are now. I was there November 20th through the 1st. I had 7 sampling days and 4 different sites. And all the bats that were captured were successfully released within an hour after capture. So, fishing bats are, I don't know, for those who have worked with bats know this, but those who haven't, bats are very tricky. They don't like the rain, or the wind, or the moon. So there's a lot of factors that have to be taken into consideration. And with the fishing bats, where we were working, we had to take into consideration the tide. So there were often times that we were like this in water, which was a lot of fun. Lots of stories, lots of photos. There I am working on the mist nets, getting a bat out. Lots of fun, lots of good stories. Here are my methods. So the measurements that I took for the wing loading were mass, wingspan, and wing area. So the wingspan and the wing area I took using this program called ImageJ. So what you can do is you can take a photo of a subject with a measurement scale in there, and you can set the scale, and then you can take the measurement, whether it's length or width or whatever, and you can take the area. So that's what I did. So I took photos of wings for 2 weeks. And so here, well, it's not. Anyway, I would set the scale to 1 centimeter, and then I would get the wingspan and the wing area. Oh my goodness. And then additionally, I took the standard measurements. And so here are my results. So for the body mass, for the males, I had a mean value of 68.6, and for the females, I had a mean value of 58.8. So that is a significant difference. That means the males are roughly about 10 grams heavier than the males. I mean, than the females. The males are heavier than the females. For wing loading, the average value for the males was 3.8, and the average value for the females was 3.5. So this is what I predicted, that the females would have a lower wing loading value, and they're not statistically different. I mean, yes, however, there's a clear trend that you can see of the females average with a lower wing loading than the males do. So results. So the males weigh more, and the females have an average of a lower wing loading. Lower wing loading, they can carry more. Why? Why? And I will tell you, it is because of the children. Okay, because of the children. This is not a fishing bat. Okay, so this is because the pups of bats are ginormous. Okay, they are like 1/3 the size of the mother. Okay, so that would be like me having a 50-pound baby. I think I was like 6 or 7 pounds, so that's ridiculous. Okay, so the gestation is at least 3 months, and then the pups fly only 6 to 8 weeks after birth. Okay, so what that looks like for me, I'm growing an 80-pounder for 3 months, and then I have to carry it around for another 2 months. So that's about 5 months-ish of carrying a lot of weight, okay? So it makes sense why the females would be able to carry more, why they would have a lower wing loading, okay? Because they have to carry the babies. So we can go back to here, right? We have the smaller bat with the lower mass. We have the bigger bat with the bigger mass. Lower, greater. And what it looks like is this. This is your female bat. It's going to have a smaller mass, okay? And the male is going to have a greater mass, okay? But the wing loading of the female is going to be lesser because the wings are greater in proportion to the body. And this works because you can add a baby onto the mother and it can still fly because of the lower wing loading. Right? So if you add, even on 200, which is like half the size of the mother, which is exaggerating it, the mother would still be able to fly. So the mother has lower wing loading, or the females have lower wing loading, meaning they can carry more because the babies are so giant. This was similar findings of the wing loading in a study done in the Gulf of Mexico. A similar trend of the female, it's flipped, but the females having a lower wing loading than the males. So a similar trend and this is within the same species, but what's interesting is that the values are like totally different. These are in the 3s and 4s, and this is in like the 15, 20s. So further investigation needs to go into this, but there's also a study done that this species has geographic variation, and this study hasn't been done in Costa Rica. It's only been done in Mexico, so there's a possibility that the species can vary, it is confirmed that the species varies geographically, but it hasn't been proven in wing loading. So that is interesting. So why bats? Why are bats important? Why do people need to study them? Why do we need to learn more? Because they're very important to the environment. Like I mentioned before, they are pest control, they are pollinators, they are seed dispersals, and they're important to the economy because they save more than $100 billion each year in pest control, in crop damage, etc. And that's it.
We have time for one short question. Richard.
Did you actually, well, you saw a lot of noctilio. Did any of them, or any of the females carry young?
Yes, there was one that was carrying the youngest. It was actually, there was a photo in the previous presentation. The last photo was pictured as that female, yes.
Because they shouldn't have to do that.
I think so too, right? They shouldn't have to carry it all. That's a lot of weight.
Bats leave their young, loose.
Yeah.
So that, they don't have to deal with all of them carrying that weight around.
Right. No, I totally agree with that. But, I don't know. It's how it works. So.
Just a quick clarification. When you said they shouldn't have to do that, do you mean like sort of, what do you mean by that?
But they shouldn't.
Probably. What? What you mean by that is that the fishing bats should be able to leave their pups in a safe place. Not that this is unfair, or whatever.
No.
So, okay. Okay.
Okay, thank you very much.
There's a break, there's refreshments, but anyone who's going in the next session and the following session, please make sure that your presentation is on my computer, okay?
Hi again, everybody. We're running a tad late, that's okay. This session is about creatures of the Pacific, and our first speaker is Rachel Davis, who will be telling us about deal vertical migration and zooplankton.
In the quiet coastal waters of Cartagena Keel, Costa Rica, a subtle but profound disruption is unfolding beneath the waves. My research focuses on the effect of artificial light at night on the nightly upward migration of zooplankton in the water column. Zooplankton are tiny marine organisms who play a pivotal role in sustaining ecological associations beneath the waves due to their role as both predator and prey to many other organisms. My study, which delves into the coastal waters of Cartagena Keel, addresses a growing concern: how artificial lights along our coastlines are continually disrupting delicate marine processes conducted by organisms, including zooplankton. The proliferation of coastal developments and fishing operations in recent decades has brought with it a surge in the use of artificial light at night in these areas, which is a significant form of anthropogenic pollution. The data collected in this study reveals the effects of artificial light at night on some of the ocean's smallest inhabitants. Every night, zooplankton undergo the largest daily migration of biomass on the planet, ascending through the ocean depths in a vital process known as deal vertical migration. By using the absence of daylight as a tool to remain inconspicuous, zooplankton are trying their best to actively avoid predation, in the same way that you try your best to actively avoid Frank when he starts growling at you. The environmental significance of DVM extends to a much broader scale than is widely recognized. This is because DVM contributes directly to marine carbon sequestration, making it an indispensable daily occurrence on Earth. This is because the ocean is commonly referred to as a carbon sink, due to the fact that it absorbs more carbon than it releases back into the atmosphere. It has been suggested that the timing of deal vertical migration by zooplankton depends on both exogenous light cues and endogenous rhythms. Exogenous light cues refer to any source of lighting external to an organism's body, which usually has the potential to influence their behavior. Just like humans wake up when it is light outside, zooplankton begin migrating upwards when it becomes dark. On the other hand, you can think of endogenous rhythms of zooplankton as you think of circadian rhythms in humans, both of which contribute to the clarification of the beginning and the end of essential occurrences, such as sleep in humans and DVM in zooplankton. Previous research hints that artificial light at night, hereon referred to as ALAN, disrupts the daily migration of zooplankton in environments such as 200 meters depth in the Antarctic Ocean. Yet similar research has a lack of being conducted in tropical waters such as those in Cartagena Keel. These gaps in knowledge lead to my research question. How do artificially lighted coastal marine environments in Cartagena Keel affect the process of deal vertical migration by copepods? My research focuses specifically on copepods, small crustaceans who form a substantial part of the zooplankton community. This also analyzes the effects of artificial light at night on specifically copepod migration in Cartagena Keel. This is important because successful DVM directly contributes to marine carbon sequestration, since it allows for the transport of particulate organic carbon from surface waters to depth. When POC is transported to depth, this leads to atmospheric carbon sequestration, an essential daily occurrence on Earth. So tonight, while you're all migrating over to Bar Amigos, zooplankton are migrating upwards in the water column, aiding the ocean in absorbing 30% of the carbon dioxide emissions from human activities, significantly dampening the impacts of climate change, which of course is almost as important as taking chili guaro shots and dancing to reggaeton. In Cartagena Keel, I chose 2 study sites, one with significant amounts of ALAN, the fishing dock, which is brightly illuminated due to the presence of large fishing vessels and industrial developments at this site. On the other hand, at Bajo Rojo, a rocky island in the middle of the ocean, the absence of daylight is not directly counteracted by harsh artificial lighting. The stark difference in light levels between these two sites provides a model to explore how Allen affects marine organisms, specifically copepods. To conduct my study, I conducted 2 plankton tows in the bottom and the surface waters at both sites. The first bottom tow being at 5 meters depth along the shallow bottom, and again at 0.05 meters depth directly underneath the ocean surface. To prevent the contamination of plankton nets in between water depths, the nets were closed using a weighted rope pulley system to close the nets at the desired depth of the collected sample. These tows were collected twice per day in dark and light conditions at each of the 2 sites, resulting in 8 total collections per day, and were conducted every other day during the 2-week study period. At the end of each collection day, plankton vials were sorted and refrigerated for subsequent analysis at Casa Verde. A dissecting scope was used to count, record, and photograph each copepod seen for subsequent identification. During the day, no significant differences existed between bottom and surface measurements at individual sites. However, the fishing dock has an entirely higher abundance of measurements among both depths at this site than does Bajo Rojo. This may be due to increased concentration of food sources at the fishing dock. You see, phytoplankton, upon which zooplankton prey, are present increasingly in number at shallow coastal environments due to the ability of sunlight to penetrate the entire water column in such locations. Tidal variations and patchiness may also have to do with why the fishing dock exhibited more overall copepods. However, the most significant data in this study was observed during the night. At the fishing dock at the bottom, there were much higher abundances of copepods than in the surface waters. This indicates a suppression of DVM by copepods at the fishing dock, which is artificially lighted. In stark contrast, Bajo Rojo exhibits extremely high levels of copepods in the surface waters as compared to the bottom waters, indicating successful DVM activity by copepods at this anthropogenically unaffected location. Conducting research on anthropogenic disruptions to DVM is crucial due to the largely unrecognized role that zooplankton play in mitigating the impacts of climate change. When zooplankton move to shallower waters at night, they feed on the abundance of phytoplankton present within the surface waters. Phytoplankton are single-celled photosynthetic organisms which convert atmospheric CO2 into organic molecules essential for their growth. When zooplankton feed upon them each night, these organic molecules burst out of the preyed upon cells, creating an oceanic pool of particulate organic carbon. This particulate organic carbon then sinks out of the phytoplankton cells, which were eaten by zooplankton, and is buried beneath sediments in the ocean floor, where it is then sequestered as organic molecules. So this whole process effectively transports carbon once present as atmospheric CO2 to the seafloor, and this is termed the biological carbon pump. By viewing DVM on a broader scale such as this, it becomes easier to understand how indispensable this process truly is. As the results of my study suggest, Allen suppresses DVM by zooplankton, which risks the integrity of one of the most effective mitigation strategies of climate change by the ocean. As we continue to inhabit and develop coastal areas like Guajira, it's important to recognize the disruptions that anthropogenic activities have on locations such as this. Through the use of artificial light at night, we are actively disrupting some of the most vital occurrences to mitigate climate change on the planet. These occurrences are natural, take no effort, and thus we should try our best to not interrupt them. This research directly shines a light on the interconnectedness of all life beneath the waves and highlights why it is important to be aware of our anthropogenic disruptions to living organisms.
We have time for questions.
Emma.
How did you first get interested in this topic? Like, what were you doing in research?
Sure. So I came here to go to Kiel, and then I'm typically have been interested in kind of organisms that aren't widely recognized. You know, like whales and sharks and stuff get a lot of attention, but things like the biological carbon pump are really like the backbone of marine food webs and processes, sequestration processes in the ocean. So I think that by acknowledging small organisms like this and recognizing the impact that they have on a larger scale, I think that that's important because a lot of people are unaware of it. Millie?
Did you notice a difference in overall abundance in the zooplankton between the two locations?
Yeah, so the fishing dock, overall abundance, spanning bottom and surface measurements, had a higher abundance, which I predict may be due to the presence of increased phytoplankton there because of the shallow, continuous nature of the benthic surface there. The sunlight can penetrate through, making more phytoplankton hang out there, making zooplankton possibly want to go feed on the buffet of phytoplankton over there, whereas Bajo Rojo drops off more steeply into the middle of the ocean and is less continuous bottom, which may contribute to the fact that there weren't as many zooplankton there. However, it's really important to note that the stark differences between the bottom and surface levels at Bajo Rojo were much more profound, which indicates the successful vertical migration by copepods in that area. Patrick?
Thank you for the presentation. My question is, you said that the migration occurs at night.
Yeah.
So why did you choose to sample also during the day? It was as a control or?
Yes, it was kind of as a control. I was more interested in the nighttime measurements, but I wanted to see how those differed between the daytime. So if they were the exact same and I only had taken nighttime measurements, I wouldn't have been able to see that in the day the bottom and surface at Bajo Rojo were like a little bit like similar, and then at night there was this stark difference. So if I didn't take those control measurements, I don't think my data would have been as meaningful. Edit?
Very illustrative figure, nice presentation. Do you know, closer to the coast, the depth is more shallow, right?
Yeah.
Do you know until which depth is this process happening? Or can you say that it's around in all the oceans, or there is a given limit?
Yeah, so previous research, it illustrates this process in 200 meters depth of open ocean in the Antarctic Sea. And my research was meant to see if this occurrence still persists in shallow tropical waters. So I picked a shallow bottom of 5 meters for each bottom surface toe, and the fact that differences still exist within those measurements illustrate that the effects of Allen on DBM by zooplankton persist in shallow waters.
Thank you.
Thank you, Rachel.
Thank you.
Our next speaker, Michaela Gunther, is going to tell us about sea hares.
Hi guys. Like Frank mentioned, I'll be discussing my research on the active chemical defense in sea hares, specifically ink and opaline release, which we'll get into. So starting off, what is a sea hare? So sea hares are gastropod mollusks. They are in the family Aplysidae, and they are found in shallow and temperate waters all over the world. They feed exclusively off of red and green algae. And some of their common predators include anemones, crustaceans such as lobsters and crabs, and fish. And sea hares do not have an external shell to protect themselves from predators, so instead they employ a variety of passive and active chemical defenses. They're super cool. So some of the passive chemical self-defenses include their unusually large size for sea slugs. They're really big, and they're very cryptic, so they're very difficult to find. They blend in really well with their environment due to the algae they eat. It gives them the coloration. And then the algae also accumulates in toxins in their flesh, which makes them largely unappetizing for most predators. So they aren't predated on by a lot, or very frequently. And then in addition to these passive chemical defenses, they also employ active chemical defenses, which is kind of the focus of my research, through behavior known as inking or ink release. So here's a picture of the sea hare. The species I focused on that is found here in Costa Rica is Dolabella auricularia. On the right side over here is its head, which is distinguished by two ear-like projections called rhinophores. In the middle is a little hard to see. This is the mantle opening where water enters, and then in the back, on the butt, there's this little thing here that is the siphon, which is where water and then also ink and other secretions can be released out of. And then more about this active chemical defense. So inking actually consists of two different secretions, ink and opaline. So these are both produced independently in two different glands and then typically mixed in the mantle before being released out to the attacker. And this mixing is important because it activates compounds such as hydrogen peroxide that are aversive to predators. But interestingly enough, I observed in my preliminary observations that some sea hares weren't releasing just ink or the mixture of ink and opaline, but also just opaline. And I distinguish this because ink is a bright purple color, whereas ink is bright purple, whereas opaline is white and highly viscous. And previous studies have found that the ink is aversive for anemones, resulting in tentacle contraction, but it induces feeding in crustaceans, and opaline induces feeding in both anemones and crustaceans, which sounds counterintuitive, like why would they want feeding to occur when they release this ink? But it's actually a super unique process known as phagomimicry, which has only been observed in crustaceans predating on sea hares. But it's basically where these secretions distract the predator and overwhelm its sensory system long enough that the sea hare can escape unharmed. And there's a lot of research lacking on this mechanism and what secretions, how they affect these predators in this way. So my observations led me to my central question: is there a difference in the active chemical response of sea hares between the seafloor and at the surface? So to conduct my experiment, I did it at Bahia El Achal in Juaniquil, Costa Rica. Up there's a photo of it on the right during the daytime. My data collection period occurred on December 1st. Between approximately 9 and 11 p.m. So we conducted our data collection at night because this is when sea hares and the predators are most active out and about grazing. And I have a little diagram illustrating our methods we used. So the ground treatment and the surface treatment were both conducted the same way. The trials were the same. Just once a sea hare was located, the snorkelers would go down, pick it up with one hand, and bring it to the surface for observation. Whereas the ground treatment, scuba divers would stay down there and conduct the trial without picking the sea hare off the ground. The trial itself consisted of gently pressing one finger on the back end near the siphon of the sea hare, while another person would record the entire process. And the person would press on the sea hare every 10 to 15 seconds until inking ceased. And so the trial started upon initial contact and then ended when no secretion was being released anymore. And here's a photo of what the inking actually looks like. As you can see, here is the siphon in the back end. I think that's Emma in the back with her light. And so this is a purple cloud of ink, as opposed to if it was just white, it would be just the opaline. And then upon reviewing all the footage, I recorded various parameters, including the time until first secretion, the total number of secretions per sea hair, the intensity and type, and the overall duration of the inking period. So a lot of factors going on. The first part of my results, this is just dealing with the timing of the secretion. So on the x-axis we have the treatment, surface, or ground. And then on the y-axis of the first graph is the average total secretion time, or total amount of time that any type of secretion is being released, averaged for the two groups. And then on the right graph is the average time until the initial secretion, again, averaged between the two treatment groups. And then I found p-values of 0.394 and 0.984, which indicated that there was no difference between treatments in relation to the timing of the inking. This was interesting because, well, it just could mean that this isn't really a variable in the inking behavior seniors. And this could also mean that it could be related to each sea hare's inking threshold. Since sea hares living in different environments have been found to have different thresholds. For example, if one lives in a higher turbulence environment, they'll have a higher threshold than a sea hare living in a really calm environment. But since I collected all of them from similar locations in El Chal, this could explain why there's no difference. And then, yeah, that's basically what I was just talking about. Similar length and duration of inking, regardless of treatment, was what I found. The second part of my results dealt with the secretion number, each trial, compared with the intensity, which is scaled from 1 to 5, 1 being very little ink and 5 being the most amount of ink I observed. So each of these graphs is for an individual trial, so there's 4 sea hare graphs up there. I found 4 sea hares inking at the surface and then 3 for the ground treatments. And as you can see, the surface ones, these 3, oh, and the purple is the ink, gray means opaline. So you can see that in these first 3, there's a lot of opaline in the later half of the secretions, whereas the ground was predominantly ink. Only 2% of the total ground secretions were opaline, which is really interesting. And some reasons for this could be because the species I observed is a sedentary organism. They don't swim, so it would be more realistic for a predator to capture them on the ground, which could be why they release more secretions overall. Another interesting potential factor for the increase in opaline in the surface treatment is the perceived predator type. So if a crustacean were to approach and attack a sea hare, it would pick it up off the ground and bring it to its mouth. Whereas if a sea hare was predated on by an anemone, it would simply be walking on the ground and get caught by the anemone. And if you remember, opaline is a feeding inducer in crustaceans, but phagomimicry can help them escape. It could be a benefit to help to release more opaline if a crustacean were attacking a sea hare. Whereas if it released opaline when it was caught by an anemone, that wouldn't really help. It would still be stuck in getting predated upon. So this could explain why there's a much higher occurrence of ink versus opaline on the ground treatments. So these findings suggest that the perceived predator type could influence the secretion type. And this is supported by previous research that found that each gland is under independent neural control, so there is the potential that they can select and kind of tailor their defense based on the perceived predator type. In conclusion, there is little to no difference in the timing, again, of the secretion response, but the more opaline release at the surface suggests that perceived predator type could be a factor, and more research definitely needs to be done on the effect of each secretion on different predator types. And then thank you to everyone who helped with this project. Yeah, that's it.
Time for 2 questions.
Was the ink and opaline sticky at all? Like, is it something that gets on the prey, or is it something that just gets in the water?
Yeah, so it's very sticky, especially the opaline when in contact with water, it becomes super viscous, so that kind of hangs in the water column. And yeah, on crustaceans, it can kind of clog their antennules and make, perception hard. Yeah.
Did any of the sea hares start inking while you were bringing them up to the surface, or did you have to rub them to have that happen?
I believe only one of them inked initially when we picked it up. And it was actually interesting because this individual was mating, so I think that's why when I picked it up, yeah, and it released a lot more ink. So that also, was a slight variation. So in the future, it'd be best to have a much larger sample size than what we had to work with. But yeah, the other ones took longer. We had to be at the surface for a while before initial secretion happened.
Okay.
Yeah.
Okay, thank you, Michaela. Our next speaker is Jake Beckley, and he's going to be telling us about octopuses.
Good morning, everyone. My name is Jake Beckley. This is color change in octopus vulgaris. So while I was observing octopus, I was amazed with how quickly and rapidly they're able to generate patterns and colors, but also by how they seemed curious about who I was and what I was doing living around their home. This led me to the questions of what color changes do octopus demonstrate in response to stimuli, which stimuli are more successful in eliciting a response, what other behaviors manifest with those stimuli, and what advantages do those behaviors offer. I'll answer these questions by looking at identifying chromatophore behaviors associated with the stimulus, examining both common and rare color changes and patterns, categorizing other behaviors when presented with the stimuli, and then interpreting the significance of those behaviors. So what are chromatophores? Chromatophores are seen throughout all the cephalopods, the squids, octopus, and cuttlefish. They are a type of cell that is a membranous sac filled with a pigment that can be either red, black, brown, white, blue, or yellow. They're controlled by radial muscle cells, which when contract, inflate the sac and let the color show through. Each cell is linked directly to the brain, connected by nerve fibers to the chromatophore lobe, which allows for instantaneous pattern generation, faster than the blink of an eye. So at the site LHL in Coahuila, I was free dive to find individual octopuses. I would look for newly broken shells and peek around in bowls under corals, rocks, and in the sand in the hopes of finding an individual. When I did, I would set up a GoPro, a stimulus, and a nearby buoy, and then I would leave. The different stimuli I would use included a small cosmetic mirror, a GoPro, a puzzle that was a ball with flax cut inside, and a food incentive, which was clams, and then also my hand. So now it's time to look at the different chromatophore behaviors, and I identified 3 different camouflage types and 1 different communication type. So the first instance is called background camouflage. It is defined as an adjustment of color to better match the substrate that is linking. This individual, which is right here, is in his hole, and he's matching kind of the color of the sand surrounding. And you'll notice when he comes out, he matches the color of the rock behind him. Background camouflage in specific happen very slowly, and it's easier to see, which is why it is sped up. Disruptive camouflage is defined as a rapid change from background color to a pale white color, and it breaks up the silhouette of the animal, which is a way to evade potential threats, also responds to just getting too close or an unfamiliar stimuli. It happens really quickly, and it's usually short-lasting, and they return to their previous color. Deceptive camouflage is when the animal appears as an inanimate object such as a rock, coral, or the sand. This behavior appears to be the most conserved of all concealment types, and I only saw it a couple of times. So see that this individual comes out and starts observing the puzzle. Meanwhile, he's trying to look like the sandy bottom with some white spots and darker spots in between to break up his figure. He can also manipulate the texture of his skin through special cells called papillae, which can be raised to manipulate texture like it does. This particular one was really interested in the camera. And lastly, we have signaling, which is a form of communication. This is called bilateral signaling, as it only changes one half of its body, and it is a form of intraspecific communication. This response was only elicited by the mirror as a stimulus, and because of this, you can infer that they cannot recognize their reflection as their own. This individual is right here, and then you'll see that slowly the left half of him changes while the right side stays white. Alright, so the results. This table shows the total number of times behavior was shown and across how many individuals that was shown. Disruptive and background behavior are the most common and they are shown by most of the subjects. Deceptive and signaling behaviors were the most rare and shown by only a few of the subjects and are not a very stereotypical response. No reaction behaviors are times when a subject showed no change in color, which is still significant because it is a behavior, just not a change in color. So this figure shows the amount of times each stimulus evoked a certain response. So the mirror was by far the most responsive stimuli, and it elicited all 5 different kinds of behaviors. It received the most amount of no reaction stimuli or behaviors, likely because octopus are highly asocial, and at the sight of a conspecific they chose to hide. And it was also the only stimulus to evoke a signaling response. The camera, as a stimulus, incited the most background and disruptive behaviors. It was likely because there was a flashing red light, and that was enough to evoke a repeated response. The puzzle had a fairly uniform distribution of behaviors, but showed the most amount of deceptive behaviors, likely because the individuals had to come out of their den to interact with it. And the hand was not successful in eliciting many stimuli, but was useful for seeing other behaviors, non-related. This graph also shows the effectiveness of the stimuli. You can see that the mirror is the most successful, followed by the camera, and then the puzzle and hand are the least successful. This chart looks really confusing, but it's not that confusing. It's a time progression chart. It shows behaviors as the interaction progresses. So the y-axis is the amount of time that the interaction was, and each mark or data point was a behavior shown at a specific time. I made 11 of these, which would be a lot to show, so I just picked the one that showed the most stereotypical patterns. And one thing you can see is a short stop in reaction time after a new stimuli was presented. This can be seen as a jump between data points. So when the hand was presented and when the camera was presented, they stopped reacting for a bit. Another pattern is that if you draw a line across the middle, you will see more behaviors in the second half of the interaction than the first half. Both of these patterns show how they generally learn about a foreign object, judging the object as a threat before they give away their position by changing colors. Slowly they go from being cautious about an object to being curious about it. And the ability to change patterns quickly and hold others for longer is important for the ability of an octopus to constantly match its background, communicate, and evade predators. This can be seen by chronic and acute patterns. Chronic patterns are held for longer and can be seen by jumps between data points, and acute patterns are short, rapid bursts of behaviors. This can be seen by anywhere where it looks like a straight line. Here, here, here. And there's usually the pattern usually is a chronic behavior or chronic pattern followed by acute patterns. So right here you have chronic pattern and then some acute patterns. Same thing right here, right here, right here. It is also important to note that each individual's response was very varied. And these patterns aren't strict rules that they follow, but just general patterns for what I saw. Some other behaviors unrelated to chromatophore use, where they held the same color throughout the interaction. There was quite a bit of them, although I'm only going to be focusing on striking, barricading, and problem solving. Striking is defined as looking like a punch, but they don't have a fist, so it's not a punch. While it was eating clams it got from the puzzle, there was this fish that was bothering it, so it struck at the fish. It is likely that it had encountered this situation before, but since the fish posed no threat to its meal or safety, this is likely just a retained behavior. You can see right here, he's eating a clam, and then this fish swims by, and damn. He did this twice. Barricading is defined as using rocks, shells, or pieces of coral to block the entrance of their den. They do this using their suckers to manipulate an object. The biological definition of tool use is the exertion or control over a freely manipulable object, a tool, with the goal of 1, altering the physical properties of another medium, or 2, mediating the flow of information between the tool user and the environment or other organisms and the environment. By this definition, barricading is an example of tool use in octopus, which has evolved convergently to vertebrates and shows advanced cognitive capabilities. So this individual comes out of the den, grabs a couple rocks, heads back up into his den, and then uses the rocks to kind of close himself in.
Good job.
The last behavior that I'm going to talk about is problem solving. It's only occurred one time out of the 5 times it was presented to an individual. And the puzzle had flaps cut and rocks and clams inside to help weigh it down and add complexity. The octopus used its suckers to hold the ball in place while also feeling around inside the puzzle to pick out the food. And then after it was done getting the clams out, it gives the puzzle an extra pat down to see if there's any more. So you can see in here how he's got his arm wrapped around a clam. He gets it out, and then he starts going for the other one. And then once he's done, he gives the ball an extra little pat down to see if there's any more. So in conclusion, octopus employ different camouflage types like background disruptive and deceptive camouflages in response to different stimuli. They communicate with conspecifics by bilateral signaling while not recognizing their reflection as their own. Chronic and acute patterns are advantageous for changing environmental conditions and prey evasion. And the mirror elicited the most color changes, followed by the camera, while the puzzle and hand were least successful in eliciting responses. The behaviors unrelated to chromatophore use demonstrate the cognitive capabilities and explain the success of octopus in their wild. And each one also reacts independently and has their own seemingly personality. A special thanks to Leo and Steven. They were a huge help in finding and identifying dens. From them I learned kind of how to do it on my own, how to take better pictures, and they're just a huge help and inspiration. The diving center, Kwahina Keel, they were very generous with their boat, chauffeuring us around all day. And the EAP instructors, especially my advisors, Frank and Emmy, for all their help.
don't really have much time, but we'll do one question if somebody would like to ask a question. Go.
So the puzzle, was it like a, where do you get that from?
Oh, the super in Kwahinikio. They had some dog toys.
Thank you.
I will be around after the presentation.
Our next speaker is Emma Toussaint, and she's going to be talking about colonization of substrates.
Okay, hi everyone. My name is Emma, and for my project, it's titled Influential Factors on the Colonization of Marine Organisms. So I want to give a little background intro. So why I got interested in this topic of research is because, like, growing up around the water and everything, I'm sure all of you have probably realized, like, anytime anything is left in the water, like, no matter what kind of substrate it is, like, whether it be a boat or a tire or a plastic bottle, like, you'll see that things collect on it. Growth accumulates on all, like, surfaces that are left in the water for any period of time. And I just found this, like, this concept super intriguing, and I wanted to dig a little deeper into it. And specifically about, like, how marine organisms decide if it's, like, a decision to colonize different substrates. Is this a random process? Do they exhibit a preference towards substrates? And then I also wanted to go a little deeper is past substrates, is there a preference for depth? And so this is not. So my question is, how will taxa richness and diversity differ among substrates at different depths? And so in order to figure out the answer to this question, what I did, first let me give you a little background on just sort of the general process. The general process of colonization and the accumulation of a biofilm on substrates that are submerged in water. So as you can see here, this is a little like timeline, and so this is the submerged material, and it goes like immediately by minutes, particles of organic matter start colonizing, and then you have primary, secondary, and tertiary colonizers. And what's amazing is that in only days, secondary colonizers have already arrived at the substrate, and in weeks, and in the 8 days that I performed my study, tertiary colonizers also colonize the substrate. And this includes, the tertiary colonizers include like macroinvertebrate larvae, macroinvertebrates, all types of algae, cyanobacteria, and things like that. And then if you progress for months, you get into the more macroscopic level. Okay, so to investigate this question, what I did was I created an experiment. I created these structures. They're sort of funky looking, as you can see right here. So basically, what I wanted these structures to do was stand vertically in the water column. So what I did was I collected 9 really big rocks, and then I sort of wrapped this fishing net around it, and then attached to it this column of like plastic mesh fencing. And what I did was 3 meters by 10 centimeters. So if you can imagine, it was a pretty skinny, tall strip. And at these heights, I attached at 3 heights 1 meter apart each. I attached different substrates. And then this column was held up attached by a water bottle floating. And so the different substrates that I placed were coral rubble and plastic water bottles. And then I left, so that I made 3 structures with coral rubble, 3 with plastic water bottles, and then 3 I left with no additional substrates attached, just to see how they would react to a control. And what I did was I placed each of these 3 structures, I placed 3 at each of these sites. This is the Bay of Kohina Keel, so we have Octopus Garden, LHL, and Baja Rojo. So to get some variety in the sense of like protected bay versus more open ocean. And so these are sort of the initial results that I discovered. So I have a Venn diagram. This doesn't make much sense right now, just these plain letters, but I'll explain. So what I did was after leaving these structures, these 9 structures in the water for 8 days, I observed, I collected them and took pictures of all of the growth that had grown on these structures. And I morphotyped them, and so that basically means just like identifying like what you can observe as like morphospecies, like miniature species sort of. And so I was interested in where was each morphospecies located. And so these are my different variables. I have plastic, coral, and control. And as you can see, there was a preference towards like plastic. Only these morphospecies were found on the plastic substrates. Only L was found on coral, and then only these were located on plastic and coral. And you can see there was only 1 morphospecies that was located at the control, and then there was a lot that were found among all 3. So here I'll get into little pictures. These are the plastic ones. So these are some of the things I found. This is called a Bryozorio, and it's basically a colony of organisms. These are actually animals, they're predators. This is a colony. This is brown algae. This, I don't know if you can see very well, this is actually a sea hare. It's called rickets sea hare. We found crabs and then all sorts of other growth. This was on the coral. This was the only unique morphospecies to coral. This is a kind of green algae. And then we had also on the control, the only unique species to control was this sort of tube-like growth, as you can see right there. And then on plastic and coral, we had a few overlapping species. This is a type of crab larva, and this is also a different species crab larva. And then we had some green algae growth, and some of this red spots we haven't been able to identify. But these are the red and green algae that we've seen are cyanobacteria, which are super interesting because they're some of the earliest life forms, the first to produce oxygen. And so it's really neat that we just can just easily see that growing on our structures. And then here we go. This is getting into what I saw growing on every structure. So this is all the overlapping. We saw lots of things. Here's red algae. These are hydroids. And these little star-like bursts are polyps, and they're used for eating. So they're actually animals, which is so interesting. People wouldn't realize. And then these are the rest of all the different morphospecies that I found among all of the structures, all the substrates. Okay, so results. This is a table of the average number of morphospecies present at each substrate. So as you can see, coral had the highest, and then plastic, and then the control. There is no significant relationship, but it is interesting that coral had on average the highest, and also that there were certain species that only colonized on coral. And then I did the same thing for morphospecies but present at each depth. And what's interesting is at 0.5 meters depth is where there was the most average number of morphospecies. And there is a significant relationship that I found between depth and number of morphospecies present. So that means at the shallowest site among plastic, coral, and control, the most species were found. And so basically, just a little bit more about what this means is that there should be consideration when thinking of colonization, especially in terms of like artificial reef, like a consideration of depth taken into account. Because as you can see, there's a relationship of more amorphous species present at shallow depths. There could be a few reasons for this. Some could be like greater access to sunlight. Another reason could be at the bottom, I observed more fish eating the amorphous species that were there. For example, the one that we saw in Jake's octopus video, that was a very frequent one at the bottom. And so yeah, and just in conclusion, basically, there should be a lot more research looked into this, just because this process is like the fundamental building block of like all growth in the ocean. Like this is where it starts, and especially like with climate change and increasing water temperatures, this process is going to be affected. And so any more research that we can get into it would be amazing. And lastly, acknowledgements. Just want to thank everyone who helped on my project.
We have time for several questions.
Why do you think that more species and more types settled on in the shallower waters?
So some of the reasons I think is because like it's since it's higher in the water column, it's easier because like a lot of ways that these organisms disperse is just like floating. It's sort of like how here we learned about like dispersal by wind, but just in the ocean, so it'd be higher chances for them because there's more space up there. And also a few of the other reasons I mentioned, like greater access to sunlight would promote like quicker growth in this short study period. Yeah.
Do you think that there's more growth in the shallower waters because of runoff in those reef areas?
Yeah, that definitely could be a factor, a potential factor. I could see. There was definitely also interesting that you mentioned that because the growth varied a lot between sites in terms of like, for example, Octopus Garden, which was closer to reef runoff than, for example, Bajo Rojo, and there was much more growth on those structures overall. Yeah.
Nice story. Could you explain a little bit more about the controls? I didn't get what is the control and what is effective to compare with others?
Yes, so the control, my idea behind that was just to make an identical structure to the ones I had, but without the presence of like additional substrates. And I was able to compare that with the ones I had found. And like, for example, an interesting result was like, there were no macro invertebrates at all found on any of the controls. And so a reason for that might be the type of substrate or how the substrates sense that like additional presence of like a hiding spot might have been something for them. So yeah, there was distinct differences between the controls and then the added substrates.
Thank you. Our next speaker is Rin Hemegren, and she will be telling us about interactions among frigate birds. Good. Is that it?
So I am going to be talking about kleptoparasitism in Frigata magnificens, commonly known as magnificent frigate birds. And more specifically, I'm going to be looking at patterns in sex and age class of the pirates. So actually before I do that, I'm going to do some background. So kleptoparasitism is a foraging behavior where, the one animal will steal either food or other material from another animal, and the animal that does the stealing is known as a pirate, and the animal that is getting stolen from is known as a target. Frigate birds practice kleptoparasitism. They can be seen often targeting other seabirds such as boobies, pelicans, and gulls, also other frigate birds. Their agility in the air allows them to maneuver and pull wings or tail feathers to knock a target out of the sky or out of their flight path and regurgitate or release their food. So in my research, I aim to understand and describe the circumstances and patterns of kleptoparasitic chases, both successful and unsuccessful. And from there, I ask the question of which individual assumes the roles of target or pirate in these chases. So in my methods, to do this, I wanted to do supplemental feeding time. So I would toss sardines into the air for the birds to catch, and then from there I could look and see chases. So before getting out onto the boat, I would get bait from a bait shop, the Kavaya sardines, and I would usually do either one, ranging from 1 to 5 kilos. And then once on the boat, I would prep the bait by cutting it into usually on average 4 different pieces. So that was about this size. And then we would find the birds. I wanted to make sure that there was birds in the area before I started throwing the bait off the boat so they could see it and come over, and then from there, that group would attract a larger group. So then before I started feeding, or what the feeding looked like was I would have one person throwing bait just pretty much directly into the air, and then everyone else on the boat, if they wanted to help, which everyone awesomely did, would record videos just on iPhones, and then after a given amount of time, usually from 1 to 15 minutes, I ended that feeding time but continued the recording until the group of birds dispersed or flew out of range from where I could see them. And here's a little map of Golfo Santa Elena where I did the boat, and each dot represents a supplemented feeding time. So the red is Isla David, and then the pink is Cochinequil Bay, and I will be referring to these later on. And then the data processing methods, once I was off the boat, I would just watch the videos and look at the composition of the group, and so that was species, and for frigate birds, I could differentiate between sex and age class, so sex and age class for frigate birds, and then I would look at chases and who was catching the bait thrown off the boat. Let's see. Not going to do this video. So then, of the 7 observational days, 59 chases were recorded, and a really small amount of them ended in success. Only 8 of the 59, so less than 15%. And 21 of the chases were group, and 38 were solos. And of the chase time, it ranged from 1 second to 87 seconds, and the mean chase time was 12.5, with a standard deviation of 16.3, and the most common chase time was 2 seconds. So intraspecific chases were most common. Those were 76%, or 76.3% of all chases, and then of those, male frigate birds were twice as likely to beat the targets than juveniles and females, and males were observed half as often pirating than juveniles and females were. And makes a little bit of sense because males were recorded catching over 50% of tossed bait. So here's some nice little photos of, I've got, let's see, let's see. So this is a female and that's a male. Just before this photo, she grabbed at his tail feather, and then here you can see he's got his mouth open, and he regurgitates his food, and she immediately flies out of his path and grabs down into the water and grabs it. And here's another female swooping in to also grab some. So that's what a successful chase ended like. All right, so comparing my different data, I did contingency tables and then tested these by doing chi-square analysis. So this one I did pirate, sex, and age class compared with chase time. So the F, J, and M in the columns are female, juvenile, and male, respectively. And then in the rows, I have chase times, the frequency of chases in those times. And after running the chi-square test, you saw that there was no correlation between these two things. And the next one, I compared pirate, sex, and age class and the frequency of group versus solo chases. And again, there was no correlation. And then this one was sex and age class of targets. So I've got female, juvenile, and male, and then, like I said before, I couldn't differentiate sex and age class of different species. So those are all, I've got like pelicans, terns, and laughing gulls in that column. And again, there was no correlation.